the next one, arrange the following discoveries in the increasing order. Okay, so we need to arrange the order. Okay, the first one is John Mendel discovered the basic principle of genetics. Charles Darwin published a book on origin of species. Watson and Crick discovered the DNA structure. Frederick and Michelson invented a nucleon. Oswald Avery identified the DNA as a transforming principle. So this is the order. We need to arrange in the correct order of increasing order. Okay. So the first one is Charles Darwin, which is origin of species of book. And the next one is John Mendel, the principle of genetics. And the next thing is Frederick, who has identified the nucleon. And the after nucleon, it is a Oswald Avery, transforming principle. And the last one is Watson and Crick, DNA. Okay, so the option C will be the correct answer. The order B, A, D, and E, and C. Okay. Okay. So arrange the following in order of their action. Okay. So according to the cell cycle, what is happening? Which is start from the G1 phase, S phase, G2 phase, and the mitosis cycle. So we need to arrange the order of enzyme that required for the cycle process. Okay. The first one is G1 cyclin GDA, which is cyclin dependent kinase. Okay, it is involved in the G1 phase, and the next one is S phase, where the DNA will be replicated. Okay, S phase cyclin dependent kinase, and the next one is MTF. Okay, so maturation promoting factor, and the next one is APC. Okay, which will be involved in the anaphase of mitosis process. APC CDC20. After 20, it is CDH1. Okay, so the order will be A, C, D, E, and B. Okay, the option A will be the correct answer. And the next one, identify the correct order of signaling a transduction pathway. First one, the binding of ligands to cell surface receptor, termination of signal, generation of intercellular second messenger, and the intercellular signal cascade, effector response. Okay, we need to identify the correct order of the signal transduction pathway. So the first one is once the ligands bind to the receptor, so there will be a secondary messenger will be happen. There will be release of signals. Okay, and there will be intercellular signal will be released. So the order will be ACD, and after signal it is effector response. The response has to be given to the signal. And the last one is process termination. Okay, first one binding of ligands to the cell surface receptor, and the generation of intercellular second messenger, and the intercellular signaling cascade, and the effector response, and the last one is termination of the signal. So the option A will be the correct answer. The arrangement will be A, C, D, E, B. Okay. And so the nucleoplasmic index, where the nuclear mass and the cytoplasmic mass of cell or in the state of optimum equilibrium, depends upon nuclear volume, cell volume, cytoplasmic volume, nuclear mass, cytoplasmic mass, and centriole number. So they have given the 5 different options. We need to identify which is a correct one. Okay, so from that option A and D, option A and B will be the correct one. Okay, so the nucleoplasmic index, the nuclear mass and the cytoplasmic mass of cell or the state of optimum equilibrium depends upon the nuclear volume and the cell volume. Depends upon the nuclear volume and the cell volume. Okay. And the next one, identify the inhibitory neurotransmitters from the following: glutamate, aspartate, cysteine, glycine, GABA. Okay, so from the 5, we need to identify the inhibitory neurotransmitter. It is glycine and GABA. So option D will be the correct answer. And the next one is the function of gene depends on the expression of DNA, RNA, protein, metabolite, and lipid. Okay, so it is depends upon the RNA and the protein molecules. Okay, so the DNA is a complete genome where it is going to be transferred to the transcript to the mRNA, and that mRNA is going to convert into the protein molecule with the process of translation. Only that conversion, the gene is going to be expressed. Okay, their nature is going to be expressed. So the function of gene will be depends upon the translation and the transcription process. So it will be happen with the RNA and the DNA. Okay, the option B and C. is correct. So the correct answer will be option C, which is having a B and C only. Okay. And the next one, identify the features of cancer cells. We need to identify the features. The first one, the rapid cell growth and the cell division, and the reduced cell growth and the division, the loss of normal control of cell division, reduced angiogenesis, utilize the glucose at reduced rate. We need to identify the correct options. Okay, so it is option A and C. The rapid cell growth division and the loss of normal control of cell division is the correct answer. So remaining things are not a features of cancer cell. Okay, the option C will be the correct answer. And the next one, identify the method to measure apoptosis in vitro in mammalian cell culture. MTT assay, LC, liquid chromatography mass spectroscopy, NMR, crystal violet assay, and page polyacrylamide gel electrophoresis. Okay, so to measure the apoptosis in the in vitro condition, which kind of techniques will be used? It is MTT assay and the nuclear magnetic resonance, NMR, will be helpful. Okay, the option C will be the correct answer. And the next one, identify the tumor suppressor gene from the following: RAS, RAF, PTEN, MYC, and P53. From that, we need to identify which genes are suppressing the tumor. Okay, so the option C and E will be the correct answer. PTEN and P53 will be acting as a signal for identifying the tumor suppressor genes. Okay, so the option B will be the correct answer.